Now we'll hear presentations from the 4 finalists of our Protégé Poster Challenge. The TAMEST Poster Challenge recognizes research that could have a significant impact on scientific understanding and society. The Protégés are all invited by TAMEST members to be their guests at the conference and learn more about science in Texas and how to advance their careers. The Protégé Poster Challenge is exclusively for those early career protégés and gives them an opportunity to showcase their work in innovation to research and industry leaders. We'd like to thank Shell for their support as the featured sponsor of the Protégé Poster Challenge. The judges were composed of TAMEST Executive Committee and the 2024 Program Committee. We received 40 poster abstracts prior to the conference and selected 20 to visit here in person at the meeting. We each spent a few minutes with the selected posters in our categories. We asked them to briefly explain their research, viewed their posters, and then scored them. The entities were judged on several factors, including the challenge or problem addressed by their research, the key elements and findings of the research, and the potential for significant impact on the scientific understanding in society. From the 20 entries selected to be judged at the conference, we have narrowed it down to 4 finalists. These 4 finalists were all deemed to be exemplary. In recognition of their fine work, the finalists were treated to a special networking breakfast this morning with Nobel laureate Michael Brown and the TAMEST presidency. Each finalist will also receive complimentary registration to the TAMEST 2025 annual conference in Dallas next February. Finalists will also receive a gift card from TAMEST, post-event publicity, and a recognition certificate.
Okay, so we're now going to hear a 5-minute presentation from each of the 4 finalists. After the presentation, there will be time for a couple of questions. The judges will then deliberate to choose the 1st, 2nd, 3rd, and 4th place recipients. The 5-minute time limit will be a factor in our judging, so I hope you 4 are listening to that. Stay on time. We'll announce the results and recognize the recipients on stage this afternoon. So now it's a great pleasure to introduce the finalists in the engineering category. Please join me in welcoming Paola Passalacqua, a Ph.D. from the University of Texas at Austin. Welcome, Paola.
Thank you, Karen. Thanks all. It's an honor to be here. So I'm going to be talking about flooding, and probably many of us in this room have experienced or have seen flood events, and hopefully not many of you have been caught in one. I was caught in one as a child. The thing is, these are extremely deadly and costly hazards. Just 2017 alone cost $60 billion, and probably you remember that was the year of Harvey. And so what is the issue? The issue is we get weather forecasts, we know something bad is going to come, but we can't map what flood inundation we're going to get. We just don't have tools that are fast and simple enough for that information to be available on the ground really quickly. You can run models and take a long time, but nothing is really available right now. And so this is, unfortunately, floods are gonna get worse with time. And so we set to address this challenge. And so we developed a series of open source, simple, but fast, near real-time tools that can account for compound flooding. And for that, I mean that it doesn't only flood from rivers. We're used to think about rivers expanding in the floodplain. We have flood maps for that. But we can also flood directly from rainfall on the surface. It can flood because of a storm surge, and we don't have any information on that. And so we developed these tools. Now, what information do you get? So this is an area of Austin. The UT Austin campus is part of this. What you see in colors are how much the inundation would increase just due to rainfall with respect to river flooding. So the black blocks that you see there are areas that experience only flooding from rainfall. Why does it matter? Because if you bought a house there and you look at FEMA maps, you would not expect to flood because that's flooding from rainfall directly. If you remember in Harvey, 50% of the Houston part that was flooded was not in the floodplain. So you buy a house there, you don't expect to flood, and you're submerged by water, okay? And so this is important also because we want to know who's most impacted. We have different storms. Your capacity of responding to a storm will depend on what's your history, what's your socio-demographic status, how much money do you have, and so forth. And so we combine this information with social vulnerability, and we get to know that East Austin, for example, that's where marginalized communities are, and that's actually where there's also a lot of flooding. And so we have high social vulnerability and high flooding, which means if you're into planning and you need to plan where to intervene, you gotta get to these folks first because they have a hard time responding to this event. And another way of seeing it is road access. You may be during an emergency and you have reduced road access and you need to get to a hospital. You need to get to a grocery store. Well, the point is, if all these lines indicate social vulnerability, so again, your capacity of responding to those events, if all these lines were to be on the diagonal, it would mean that that impact of road closures impacts everyone the same way. Well, it's not true. Those lines are quite far away from the diagonal, which means the burden actually of road closures is on the upper 25% of the population that is the most vulnerable. So there's an inequitable distribution of the impacts of flooding. And so I started these talks saying we don't have enough information on the ground. So what are we doing to put information on the ground? First of all, I lead a center that was just funded last year by the Department of Energy. This is a large investment that allows us to look at compound hazards. We don't only have flooding in Texas. We have droughts, we have heat, we have air pollution. If you look at Southeast Texas, there's a lot of vulnerable communities that are exposed to compound flooding. So this center is co-developing strategies for adaptation with those communities. We're co-developing them. They work with them. We work with residents. We work with industry. We work with governance. Everybody in the same room to find solutions. And finally, we put these tools right on the ground in the hands of the firefighters. There's an application that, as of 2 weeks ago, was used in the last storms in Texas here, is now operational over the state. And this allows firefighters to have flood inundation maps in their hands. The thing that a firefighter will tell you is that it's night, it's dark, they don't know how bad it is. Well, now what they can do is to drop a pin where they see water, and based on pre-compiled flood inundation maps, they have the likely extent of the flood. And this will help us save lives, save property, and improve the resilience of the natural, the built, and the human system. Thank you very much. Oh, I'm sorry.
You have a question or two?
I thought I was done. There you go. Please.
I think the mic is on. Oh, okay,
It
wonderful.
is.
So that was really fascinating. 2 quick questions. What was the greatest challenge to your work? Was it obtaining the data or putting the algorithm together to combine the data?
I'll tell you, well, of the two that you answered, I would say data for sure, because it comes from different sources and you have to put them together. But I will tell you that I think my biggest challenge has been that in academia, there aren't a lot of incentives to put solutions on the ground.
Okay.
And flooding has been doing research for decades. And then you look at a flood event and you're like, why don't we have this information on the ground? It's buried in some paper that I need to publish, right? It makes sense to publish, but it's not on the ground. So I think the biggest challenge, and I think that's where we're succeeding, and UT Austin has actually put a lot of resources into that, is into bridging that continuum between the fundamental research and the application on the ground.
And the second question is very short. How portable is this? I'm in Houston. I would love to be able to see you doing this down there.
Well, the firefighters will happen to flood. So that's very portable. You as a resident, you can download the tools and use them. No, it's for the whole state.
Fantastic, congratulations. Thank you. Okay, it's my pleasure to introduce now the finalist in the science category, Dr. Matthew Sieber from the UT Southwestern Medical Center. Matthew.
Thank you very much. It's a real pleasure to be here. My lab is interested in the basic biochemical mechanisms that produce energy and biosynthetic precursors for growth. In particular, we study an organelle called mitochondria, which is one of the major factors that drives energy production and the interconversion of small molecules in the cell. Our lab studies how corruption of mitochondrial metabolism influences disease progression as well as normal development. To do this, we study a multi-organism approach using model systems such as Drosophila, human cells, mice, and organoids. We apply basic biochemical approaches as well as systems-based approaches such as proteomics and metabolomics to examine a number of understudied questions in the mitochondrial field, including how this organelle that controls metabolism gets remodeled between cellular dormancy and cancer growth. We're also examining how small molecule metabolites that can be loaded into an oocyte and function as a heritable factor to influence progeny health and disease susceptibility. But for the sake of this talk, I'll briefly mention a story done by a postdoc, Yipeng Du, who is examining how mitochondrial metabolism can influence the identity of cells. We started this with a very simple question where we wanted to, much of the work that we know about these organelles, mitochondria, stems from how mitochondrial dysfunction intrinsically affects a cell. But we're quite interested in understanding when you have mitochondrial dysfunction in a cell, what does it do to the neighboring cell? This is really relevant for cancer because many aggressive cancers are corrupted at the mitochondrial level. But it's very difficult to study in these types of a system because there's a lot of cellular and genetic heterogeneity within the tumor itself, as well as in the surrounding tissue, there's a lot of different cell types present. So understanding the signaling roles of these cells can be quite challenging. So we started this study in a simple Drosophila system, particularly looking at Drosophila oogenesis, in which we have a case where a germ cell is developing in tight association with a somatic cell. And there's a lot of signaling going back and forth between these two cells. And we did a screen, and we just mutated genes involved with metabolism, energy production, and biosynthesis in the cell. And then looked for factors where the germ cells continued to develop normally into an oocyte, but these surrounding somatic cells displayed a developmental abnormality, or they failed to acquire their mature identity. And what we find is that normally these somatic cells here, they proliferate early in the development of an oocyte, but then they stop at this point in development. And then they mature and acquire their mature fate. However, if we disrupt mitochondrial metabolism, in particular, if we disrupt mitochondrial metabolism in a way that produces a toxic oxidative compound called reactive oxygen species, these cells fail to acquire their mature fate and they continue to grow. What we have found is the cause of this, that links mitochondrial dysfunction to this effect on development, is that the mitochondria produce ROS, which disrupts the ability of the cell to traffic cell surface proteins, in this case it's an example called delta, to the plasma membrane. And so once on the plasma membrane, these proteins can signal to an adjacent cell and activate a specific mechanism in that cell. What we have found is that when mitochondria are dysfunctional and they produce this toxic oxidative compound ROS, the receptors no longer localize to the interface between these two cell types and are trapped within the germ cell. If we simply restore normal trafficking of these receptors, we rescue the presence of these proteins on the plasma membrane, but we also rescue all of the developmental abnormalities. What's interesting is if we apply this model take this model back to our cancer systems, what we have found is that we wanted to examine how mitochondrial corruption in this cell is activating or suppressing pathways in the adjacent cell. And what we find is that when you have this disrupted mitochondrial function, you actually suppress the innate immune signaling pathway and increase pathways that promote metastasis and angiogenesis. So this would promote the aggressiveness and the spreading of tumors. And so to expand on this, we went into both human cell models as well as mouse tumor models and found that models that have severely dysfunctional mitochondria and produce a lot of this toxic oxidative compound ROS also display defects in the protein complexes that control the trafficking of this protein to the cell surface. So it disrupts the cell-cell communication in these tumor models. So overall, what we think this work provides is a context where, while most of our previous work has examined how the intrinsic effects of mitochondrial disruptions on disease progression, it also shows that there is a non-cell autonomous effect in the adjacent cells that really we should examine to see how it contributes to disease pathology. We also think this provides a mechanism for us to develop therapeutics that can bypass the effects of ROS production on these adjacent cells by simply restoring protein trafficking. And with that, I'm happy to take any questions.
I'm looking to see if anybody else is going to ask the question. So this is very interesting. Cheryl Walker over here. Real quick, do you have any data on whether or not this might be affecting in the cancer setting the immune cells looking at the tumors? Can some of the adjacent cells be immune cells?
That's a very good question. We're actually in the process of looking in mouse tumor models and mouse xenograft models to examine that question directly. We don't have any data for that, though.
Thank you.
Thank you very much.
Our finalist in the technology category is Dr. Aaron Becker from University of Houston.
Thank you. So I want to tell you a little bit of a story because I'm a landlord and I got a phone call that nobody wants to get this summer saying that their plumbing was stopped up. And so the plumber came out and there were about 3 different options of things they could do. They could go into my front yard and dig a hole, find the pipe, remove that pipe, put in a new one with no blockage in it, seal that up, and then my grass would look just as normal in a couple years. Number 2 is that they could try to snake a cable through the toilet and try to reach, if they can get around all the bends and get out there. And most of the time, they'll only be able to take out the blockage and they won't damage the pipe. And the third one is maybe we could pour something like Drano down that system and let that percolate through there, and then it'll slowly eat through the blockage. So, none of those are wonderful options, but I want to talk to you about a disease called pulmonary embolism and deep vein thrombosis. This is where you have a blood clot that travels usually from the legs, and then it's pushed, it becomes dislodged, and it goes through the system, and then plugs wherever it gets too narrow, and that's usually in the lungs, so the pulmonary part of that. And this affects, the CDC estimates about 100,000 Americans die from complications of pulmonary embolism. And so just like in my yard, wouldn't it be nice if I could make a little submarine, steer it down there, and only attack that blockage? And so that's something that my lab has been looking at because the typical treatments for pulmonary embolism is to try to put some clot-busting medication down there. And there are significant populations where that isn't a viable option, especially for our cancer victims, the geriatric populations. You can also try to snake through a catheter. And I've got a picture here from a colonoscopy. And there's a reason why people don't look forward to these. Because every time you're trying to push a tube through the body, if you want to change direction, and there's a lot of direction changes in your body, you've got to have an equal and opposite push on something else to do that. And there's some distension in there, and so you don't have to do this every year, most of you. And so what my lab has been looking at is how can we make a tiny robot? And so here's a video of one of our tiny robots. Our robot is, the thing that you see here is like a wood screw. It's only about 5 millimeters long, and we put a magnet in there. And a magnet always points toward magnetic north. We put a bunch of copper coils around these, and we put energy through there, and we can make magnetic north in any direction that we want. And we can steer this robot through there, bring it right to that, oh, it's not playing. Let's try, oops. Maybe you can make it play, but I'll tell you about it. So it's a little robot, and we can bring that to the blood clot, and we can burrow through those blood clots, and then we can navigate back. So we introduce this with a catheter, and then we can retrieve it with that same catheter system. And so the robot is not just this little piece of plastic at the end. The robot is the entire system, the electromagnetic coils outside, and crucially, the imaging system. And so we started with cameras, and we can drive this thing at 100 millimeters per second, which is 20 body lengths per second. Usain Bolt can run at his fastest for 100 meters. He can run at 2 body lengths per second. So if we could run like that, that would be amazing. And then we have the industry-leading rates of being able to go through these blood clots. But pulmonary embolism is just the tip of the iceberg because we've got this system where we can steer things around. We're now using an ultrasound that is mounted on a robotic arm so that we can steer that ultrasound and follow that through. Now I'm going to end with my ask since I'm here with a bunch of wonderful people. And so if your hospital is retiring a fluoroscopy system and you're looking for a place to donate that, we'd love that because ultrasound is wonderful for a lot of reasons, but it doesn't see through air pockets very well. And so lots of applications for impact in this area, and those, the pulmonary embolism, sort of the tip of the iceberg, because those are the biggest clots, easiest to access. But we've got a roadmap for miniaturizing these devices so we can then start to look at other applications such as strokes in the brain, interventions in the heart, and then using these same vehicles, it's a robot, where an end effector, we can make that end any size or shape that we want. And so we can use that for things like biopsy or for medicine delivery. So thank you very much.
Thank you so much, Dr. Becker. We have time for one question, please. All right. Thank you so much. So it's my great pleasure to introduce our finalist for medicine, Dr. Stephen Yee from University of Texas at Austin.
Hello. All right, so first, thanks the conference organizers and everybody for having me here. I'm really, it's a great honor for me to share some of the work that we recently did. So here, I hope by the end of the talk, in 4 or 5 minutes, I can convince you that AI and machine learning approach is really a perfect solution for this important problem. What is the problem? So this is a medicine session. So we are talking more like how our biomedical research can benefit or translate into medicine. So the current status of cancer, for example, as one example of disease treatment and therapeutics, is that patients, after they are diagnosed, there is a very standard, limited amount of standard care options available. For example, in the case of cancer, even some of the very severe, rare, and serious cancer types like triple-negative breast cancer, for example, chemotherapy, radiation therapy, and limited amount of targeted therapy are really all the options available. I was saying that I was in the morning having the breakfast. I was one time in a room discussing with some of the physicians treating patients. When it comes to a scenario that is a difficult case, the patient is really desperate, they need options for treatment, but the doctors finally say, you know what, we have one last option, is try immunotherapy. Again, it's a try, no guarantee. So here, we really hope that we can offer, which is the future of cancer treatment, some of the precision medicine options, especially giving new options, additional options that we can give in an individualized, more precise way. How do we do that? So we took an AI or transfer learning approach here. So basically, the first step is to sequence, really, the biopsy or tumor from the patients. As mentioned before by one of the previous speakers, to tackle the heterogeneity of tumor, we really need to use single-cell sequencing approaches to capture, dynamic cell types and microenvironment of the tumor better. Step 2 is really to compare the tumor data with cell line data. Why we are doing that? So that is actually a huge opportunity to take advantage of the cell model, pharmaceutical data, drug sensitivity data, drug response data from big pharma or individual labs and researchers available. So far we haven't really taken advantage of that data yet, and we want to use that for precision medicine prediction. So that's our step 3. So the idea is that taking consideration of the different proportion of cells possibly in the tumor, taking consideration of the cell response for each individual cell line, again, from the large amount of biopharma data or individual wet lab data. We can build a model there. I'm not going into details because of the time limitation. And we can calculate a single unified drug activity score. So we can rank the drugs for every single patient. So rank number 1 will be that particular combination, number 2, number 3. So essentially, we can get a atlas, the so-called patient tumor cell atlas. On the left, fortunately, I was discussing some of the poster discussion detail yesterday also. We were able to separate different patients into different colors you see on the left. They are really representing different patients. So that gives you an option. They are not really mixed up, and there's a hope that we can do something to translate that into similar cell lines, again, leveraging on the drug response data. When you see on the right, that is actually a direct mapping into the drug sensitivity data for each cell line. So you see here as one example that purple patient on the left mapped to a cluster the same embedding. You see basically the drug, so on the right, that's different drug combination color-coded. It's mapped majority to the blue combination. So, down there, that's exactly the combination. So this way, we were able to really offer some options, again, step one, for patients in an individualized manner. Again, long way to go, clinical trial and FDA-approved drugs and all of that. But we hope that can be a way to start going in that direction. Thank you very much.
We have time for one question, perhaps.
Thank you very much.
Thank you so much, Dr. Lee and Dr. Yi and the rest of the finalists. So the judges will now deliberate, and we're going to announce the winners at the conclusion of the conference in the afternoon. Thank you.